Hey guys, welcome back. Sorry, there's going to be a lot of noise going on. I have people working on it at the house. I think I repeated that as well in the slideshow. But nevertheless, we're going to move forward. We have the eyeball, okay? And we have the ear to go over. I have both models. I'm going to try and do something with the models. We'll just go over the brief anatomy. Most of it's going to be in the PowerPoint itself just because it's so difficult to do. If we were in the lab itself, you guys would be grabbing these, taking them apart, and with a partner and kind of going through and trying to label them, okay? But it's all online right now, so best that we can do is this. So let's go ahead and move on forward with it, okay? All right, guys, let's get started. Okay. So the eye model itself, I'm just going to kind of go through and take apart each aspect, and we're just going to briefly go over this, and then we're going to hit up the PowerPoint because I really feel that that's going to be the bread and butter is the PowerPoint itself, because you can use the PowerPoint in conjunction with filling out your lab assignment. Okay. Okay. So here we can see the outer covering of the eye. Really, this is the eyelid, okay, and this is muscle tissues of the eyelid. On the inner aspect of this, if I go ahead and take this off and I flip it around, then we can see on this inner portion here is the conjunctiva, okay? So if this gets a bacterial viral infection, then we have that conjunctivitis, all right, and then that can cause some redness here on our eye itself, or maybe even to the sclera, or maybe the bacterial infection can sometimes, although rare, migrate onto the sclera itself. But that's if you let it get really bad, okay? But really it starts off kind of on this tissue here on that inner part of the eye. Remember, that just sits here. And that helps provide that lubrication and helps provide the fluid from the glands to be released and pass through and lubricate the eye. Okay, right here we can see the superior rectus muscle. Okay, and the superior rectus muscle is the muscle that lifts the eye upwards. So you can kind of see it there, and also helps to control the movement of the eyelid there as well. All right. Okay. So the eyeball itself. What's this portion right here? Hey, Alex, that's the cornea. Yes, you're correct. That didn't sound creepy at all. That's the cornea. Underneath the cornea, remember we have the opening, and it's kind of hard to see because this has got all foggy, but we have the opening. So underneath the cornea, we have the opening is the pupil, and then the iris is all around it, and the iris is that basically the control of the contraction or the dilation of the pupil itself. Our sclera is here. That's that outer layer, okay, that outer covering layer. All right, let's do a little sagittal view, if you will. Okay, but only if you will. All right, so here we see a really good look of the muscles. All right, so we see our superior rectus muscle, our lateral rectus, okay, inferior rectus, okay, and then we have the inferior oblique. Kind of hard to see, but you can almost see it right through there, right, as it wraps around, and then it comes on up through here. Okay, that's that inferior oblique. And then here in the back, you can see the inferior rectus right there, okay. Superior oblique is up through here. It's kind of hard to see because the superior rectus kind of covers it from your angle right there. You may be able to see it if I tip. No, it's going to be too hard. Okay. Yeah, other than that, you can kind of see inside. See the optic nerve is right through there. So maybe this will kind of help you out, give you another sort of viewpoint of how the eye sits into the orbital cavity. Remember, the bone that we want to think of with the eye is the sphenoid bone. Okay, our sphenoid bone is here. Our maxilla here. Our ethmoid is deep on the medial aspect of it. Our ethmoid, we want to think normally more about the about sense of smell, because the ethmoid really plays a role with that cribriform plate. remember, in the olfactory foramina. But the optic nerve goes through the optic canal or the optic foramina back here, a part of the sphenoid bone. Okay. So the good old eye. Not much we can do via video, so the majority of it's going to be on the PowerPoint. But there you have it. At least we were able to touch it, and you can touch on it just a tad, and you can kind of see how it sits in the orbital cavity. And it's just kind of neat looking, you know. Too bad we're not in lab and we can't get our hands on it, okay? Okay, so what are we talking about here? Well, we're talking about the eye. The eye is one of the most important senses that we have, arguably. And first, let's go over just the basic anatomy, okay? And then I want to talk about some of the functions and basic function. We're not going to go too into detail with this, because remember, this is just a brief introductory into the anatomy of the eye and a basic physiology of the eye as well. And then same with the ear, okay? So what are some of the things that you need to know? Okay, well, we're going to go over the muscles here in a minute, but right off the bat, we need to know what the layers of the eye are, okay? And so the outer layer of the eye, kind of that white portion of the eye, is called the sclera, okay, the sclera, all right? And this kind of encapsulates the eye. It's an important structure of the eye. It's one of the outer coverings of the eye, and we really want to make sure that when we're talking about pathologies or so on and so forth, that this is kind of the first line of defense to anything really into the eye, besides our eyelids itself. The sclera is hands down one of the most important parts of the eye, as it helps to protect the inner aspects of the eye. They're a little bit more important. Okay. Deep to that is the choroid. This is a layer of blood vessels and tissue, and we're going to go into a little bit more detail with some of this stuff. But the choroid is basically how the inner portion of the eye is supplied with blood, okay, and removal of waste products. Just deep to that is one of the most important aspects of the eye, and it is the retina, okay? So the retina here is really the layer that has a lot of the cells of the eye that help us collect light, okay? And if we damage the retina, which we'll go over here in a minute, like a detached retina, that's when we can really have some problems being able to see and collect some of that light. Okay, so this is the retina here, as you can see, and then the choroid kind of goes around the entire eyeball itself. At the very front, light needs to get in, right? Light moves in, and light has to pass through the cornea, okay? And then from the cornea, it passes basically through a hole, all right? And that's what the pupil is. The pupil is the hole. And the pupil is controlled by the iris. And the iris either contracts or dilates dependent on the muscle tissue, like the ciliary body and the muscles. The ciliary body and the muscles help to pull actually relax, thereby causing the pupil to dilate or contract, causing it to constrict, okay? So once it contracts, then the pupils really get nice and small, okay? So when we have a lot of light entering in, right? We all know this by now. When we have a lot of light entering in, then the pupils get relatively small, okay? When we don't have a lot of light coming in, the iris tends to be a lot larger, so that way more light can be collected in, okay? Sorry, if you guys hear some banging noise going on, I got a bunch of people working on my house, so might be kind of loud. If you don't hear it, then good. All right, so what are some other things that we need to go over that you need to know? So you see the lens, okay? The cornea, the pupil, the iris, the sclera, the retina, the choroid. All right, and then we have the optic nerve. Remember, the optic nerve is cranial nerve number 1. All right, that's the optic nerve. Excuse me, not cranial nerve number 1, cranial nerve number 2. Sorry about that. Cranial nerve number 2 is the optic nerve. And here in the inner portion
of the eye, in the inner portion of the eye, this is called the vitreous body. And this is filled with a fluid or a gelatinous fluid substance called the vitreous humor. All right. The vitreous humor, this also can be an area where, if you've ever heard of the term floaties, or maybe you're kind of looking up into the clouds and you see some weird things kind of floating across your eye, look like little squigglies. Well, that's because they exist in here, and then they float back and forth, and then they cross the plane of light as it's going through. Remember, all vision is light is entering in. Okay, so light comes in, it reacts or it stimulates the rods and the cones in the back portion. Those are our photoreceptors. And then right here is a really important location, the fovea centralis. Okay, the fovea centralis right here is kind of the focal point. A lot of cones surround this area. All right, and then so light enters in, gets collected by the photoreceptors, and then on and through. And we're going to kind of go over that in detail here in a little bit. All right. You know, your lacrimal caruncle is here. Your tear duct is here. Okay. This helps to provide lubrication. All right, for the eye. Not like that. You get red mark all the way across the eye. But so notice this. This is going to be on your assignment. Okay, so these are just going to be A, B, C, D. Sort of this will be A, you know, and then you have to name it. All right. So that'll be part of your assignment. So go over this, know it in detail. All right, this is just another cool picture. I like this picture. If we have an exam for this lab, I might use this guy because I really like this guy. He's a really cool picture. But just another look, another angle. You can see the cornea. Okay. And then we have our iris, our pupil is the hole. Then the lens. The lens helps to focus the light in. All right. Then we have our retina, our choroid, and then our sclera is here on the outside. Okay. Pretty neat stuff. That's just a neat slide. I like those pictures. All right, so what's the innervation to the eye? Well, we know that the visual innervation to the eye is the optic nerve. Okay, cranial nerve number 2. Well, cranial nerve number 3 is ocular motor. That helps to move the eye, and we'll talk about which one specifically. The trochlear nerve helps to move the eye, and the abducens nerve help to move the eye. Okay, and if you notice, this is all kind of coming off of our brain stem. You see this area here. Okay, so remember we have our pons here, and then our medulla oblongata here. All right, so we can see where the nervous tissue comes in. Now, what does what? Well, if we want to move the eye upwards or downwards or inferior oblique, that is ocular motor. Same with medial as well. Okay, so medial is ocular motor as well. So this would be considered, if we're looking at this from anatomical perspective, this is the right eye, because this is medial over here, and the nose is here. There's my nose. There we go. There's my nose. Okay, so ocular motor are those movements right there, and we can see that as cranial nerve number 3. Okay, so inferior oblique, inferior rectus, and superior rectus all are innervated by the ocular motor nerve. Okay, so we look at the trochlear. The trochlear also provides movement and basically does superior and oblique. Okay, superior, oblique. All right. And then abducens, I think of abduction. It abducts the eye, so it moves it laterally. Okay, lateral rectus. All right, so those are the movements, and then those are the nerves. So we have 3 nerves that affect movement on the eye, and then 1 nerve that helps to collect the data that is basically gained from the light being received in the rods and cones, and then that flows on through and then goes on to the optic chiasm. Okay, we'll talk about that. Here's our optic chiasm is here. Okay, but we'll talk about that in more detail, the flow of it. So let's talk about these muscles real quick, because you need to know the muscles. Okay, so I want you to go through and I want you to label these muscles, all right? And I think that's part of your homework assignment. Hopefully you have your lab sheet open. I would behoove you to have it open because we're literally going over everything in order by what's on your lab sheet, all right? And you have to label these, okay? So pretty easy: superior rectus, inferior rectus, medial rectus, lateral rectus, okay? So that's what either moves the eye up, down, right, left, okay? And then we have oblique movements, all right? And the oblique muscles do that. We have an inferior oblique and a superior oblique, okay? And that's what helps to move our eye around. Okay, so if we move our eye around in a circle, well, then we're utilizing multiple muscles at a given time. And these muscles are highly, highly sensitive, meaning that they are very, very precise in their movement. Okay, so very precise in the movement. A lot of branches of the nerves come and innervate these muscles so that we can get really good fine motor skills with the movement of these muscles. Okay. All right. So we also have muscles around the eye. Okay. And this helps us be able to blink, right? We need to be able to blink. That's important, so we have to protect our eye, all right? Probably not going to test you on the muscles that surround the eye, but it's important to know that we have those, all right? Okay, remember at any moment you can pause it and then kind of write down some of the stuff as we're going through, all right? So you're going to need to write down, in the previous slide, you're going to need to write down the muscle name, the action, and the innervation of it, as well as identify them on on kind of a matching sheet, similar to like how the quizzes have been so far. All right. So here's a neat little slide on how light enters in. Okay, you notice how the lens helps to reflect it towards the fovea centralis, the fovea centralis. Okay, this is really, really important, the fovea centralis. That's my really good handwriting with my computer. I'm going to erase that because that looks so bad. But know that that's the fovea centralis, okay? Light enters in and is picked up by our rods and our cones. And where our rods and our cones are is basically in this layer here. All right. So in this layer here, this is where we have all of our photoreceptors, okay? And those photoreceptors are there, so we have the rods and the cones here. And then, as I said, the closer you get to the fovea centralis, or the maculae foveae, it's also called that as well. But the closer you get to that, the more cones there are. Okay, and here's a really good example. So cones pick up things on a colored spectrum, or excuse me, light, not things, but pick up light on a colored spectrum. So you notice that the majority of the cones are here. We have some outliers for our cones, but the majority of light that hits this region here, so when light hits this is mostly on a shade, on a grayscale shade. So our rods pick up color on a grayscale shade, whereas the cones pick up color on the color spectrum, and we have more of those near where the center of light is focused. Now, so what does this actually mean? Here's something that's really, really, really interesting, okay? If we have light coming in and it's really dark out, this is why it's hard to see at night in color. It's hard to see colors at night. It's easy to pick up the color red at night. It's because we have a lot of cones that are more of a reddish tint. Okay, but colors like blue are harder to see at night. or yellow. It's hard to distinguish between some colors at night. But if light is coming in and we have light hitting this area here and this area here, let's say our peripheral region, all right? So in our peripheral region, we have light hitting. Our brain will actually fill in this whole field of vision. So this field of vision, our brain is so fantastical that it'll actually fill in the color for this area here. So everything that's getting picked up here, we don't really see that in color, but our brain says that, hey, that object over here that is being picked up by light over here that's reflecting off of that object, that's the color blue, or that's the color red, and our brain will fill that in. Why? Because it's also picking it up here, and then the brain just does the rest of the job and fills it in, which is pretty darn interesting if you ask me. So your brain does a really good job of integrating the data that it receives here and creating a holistic picture as we go through and as the data goes in, and you get a conscious awareness of it. Really neat, I think. All right, so visual pathway. How does the visual pathway occur? All right, well, the right visual field, the left visual field comes in, okay? Right visual field comes in, and then we actually get information coming in and it crosses over to join up with one side. Okay, then the left visual field will pick up and cross over to the right side of the brain and join up with that. Okay, and then all of this, and you can, I'm not going to do each step by step, but you can go through the steps, right? This is the optic chiasm here. All right, crosses over at the optic chiasm. All right, and then goes on through. Eventually, this right here is an unconscious awareness, what's going on. But once that information follows that track and synapses here through into the occipital lobes, all right, in the primary visual areas of the occipital lobe, this is when we get that conscious awareness of what's going on. And this happens really fast, okay? Know these steps. All right. So pause on that last slide. Go over the visual pathway. You need to know the steps on that. You need to go through the steps on that. Okay. So go over that quite a bit. All right. Let's talk about some pathologies of the eye. Okay, because we've all had, maybe all of us have had an eye injury, but let's talk about some things. So here, this is just a good example on conjunctivitis. Okay. So we have our conjunctiva is here, and it's basically the layer that is the under portion of our eyelid and kind of connects to the soft tissue of the eye, right? And it helps to protect the eye, helps to provide lubrication over the eye so the eye doesn't dry out when we blink. And it's just a really important piece of the eye just for protection, all right? Sometimes, though, that can get infected. It's either a bacterial or a viral infection, right? We've all heard of pink eye. Well, pink eye is conjunctivitis, okay? And it's recognizable by redness here. Sometimes you can wake up and you have kind of a crusty eyelid and it's shut. Sometimes you'll get these bumps on the inside part of your eye. All right, if this gets really bad, then you want to go to the doctor. If it's viral, you usually can fight it off on your own. If it's bacterial, sometimes you can. But if you start to really have a discharge that is kind of an opaque color, not clear, or crust forming, and it's hard to open your eye because you've had it really crusty, you kind of want to go in and get that taken care of from an ophthalmologist. All right, because then that means that pink eye is really getting bad, and you don't want to have it go to the inner part of your eye and do some damage onto your retina, because then that's bad news bears. Okay, so if conjunctivitis is held off for a while, that's not good. That could lead to some problems down the road, but usually this is pretty benign if you get it taken care of quickly. Okay, so that conjunctivitis. What about some visual field defects? Now you can have a visual field defects if you have some problems with your rods and cones, or if you damage the retina. Okay, and we'll go into retinal detachment here in a minute. But field of vision defects. Now we're talking about, okay, what happens if I damage the nerve connection here, or the nerve connection here, or the nerve connection here, here, here. Obviously, you can see all the areas that there's potential nerve connections, all right? So if you get damage here to just the inferior aspect of the eye, right near where the optic nerve collects information from the rods and the cones, right in this area here, either on one side or the other. If you just get it on one side or the other, or maybe right near the fovea centralis, that's called a central scotoma, all right? And that's a potential damage there. And what that causes is you get basically a dead spot, all right? And you can't really see anything there. You can have complete loss of vision if you damage the optic nerve on just one. Then you have what's called monocular vision loss, or monocular vision. All right, I'm going to sit the next few plays out. I'm having trouble talking here. Okay. So that's in B. If you damage the area in the optic chiasm, all right, so you damage area in the optic chiasm, then you get what's called a bitemporal hemianopia, okay, meaning that you're going to have half of your field of vision, usually it's the lateral aspect of both sides, that you end up losing, okay? And then I'm not going to read off every single one of these, but you can see that if you damage the area of D, which is the one side, basically the tracts going from the optic chiasm down and back, or G, any of the tracts going to your occipital lobe, or even H, down back into the back portion of the occipital lobe. Like, let's say if you get a concussion, all right, and a lot of these from here down we see with head trauma or head injuries. Sometimes you can see this occurring with a type of neural infection, or a really bad meningitis can occur in the back portion here, and then that can affect your field of vision. Okay, but you can see how the field of vision, it can be damaged by the multiple areas here, and then what the net result is, and then here we have what is the vision field deficit or vision field loss on this side. All right. Here on the left-hand side, I put this stuff up mostly for this. Okay, this is how we do cranial nerve assessment. All right, and when I suspect someone with a concussion, and I'm going to do a field test to see if their cranial nerves are intact, I'm going to look at, number one, visual acuity. So I'm going to see if they have any of this going on. And how do I do that? Well, just a series of questions, you know. Maybe I'll cover one eye, I'll cover the other eye. Sometimes the brain can make up what's lost right here. Sometimes it can't. And I also wait. I take my time. Okay. I usually check all of these cranial nerves. Cranial nerve 2 is this right here. This is 2. All of this is what we're talking about. This is cranial nerve 2. Okay, optic. But I usually take my time. I usually wait about 15 minutes. I talk to the patient slowly, as long as there's no serious red flags, because I want to see if there's any bleeding in the brain, or bleeding or damage to any of this. And sometimes it can be slow to occur. Okay, sometimes it can be very fast. If it's very fast, you're calling 911 really quickly. So that's visual acuity. Pupillary reaction. This is our ocular motor nerve. Okay, I want to see how well the pupil is adjusting to light coming in and out, or if it's tracking, or if it's moving in the same way or fashion as the other eye. And then I do a tracking test, okay? And we can talk about that tracking test here in the next slide. All right. So first, let's go over this right here, because this is the tracking test that's going to test the cranial nerves that I want. Remember, ocular motor, trigeminal, and abducens. Okay, that's what we want to look for, and those are the tests that we do. So I'll say, hey, follow a pen. It's also done on a sobriety test, because this can be kind of impaired if you're under the influence of drugs or alcohol. But a concussion and MTBI, this is what we test as well, because we want to make sure that these are intact. All right, these cranial nerves are intact. If these cranial nerves are not intact and it's due to an acute injury, then we can have one of the following here, okay? Estropia, exotropia, hypertropia, hypotropia, okay? Now, sometimes people can be born with this, all right? And if they are, then, you know, there's things that you can do. Sometimes a doctor will go in and release the muscle from one side of the eye to help straighten it, okay, in one direction, or they'll tighten the muscle in the other direction, you know, so on and so forth. They can do that. But if it's an acute injury, so like an MTBI, this is very, very serious. If they have this and they normally don't have this going on, then this means that one of their cranial nerves is not intact and not working properly. So this is an immediate call because there's a potential for a hematoma, either subdural or deeper than that as well. So you want to get them to the ER as fast as possible. Yeah, so this is called the cover test. Okay, sometimes this can occur where the eye will actually adjust and fix itself based on what's going on. That noise is getting loud. Okay, so that's it for the eye. I just like this picture a lot. Seeing the light come in. Okay, there's a ton of pathologies with the eye, but I just wanted to introduce you to those. You're more than welcome for your assignment to look up any pathologies you want and kind of write about and go from there. But then you'll have to do a visual pathway description and a visual pathway drawing as well. Okay, so that's the eye. I may pull out the model and edit this together with the model. I got some models from UNCW, but it's kind of hard to do these models here because there's so many pieces to them. It's not like the muscles or anything like that. The digestive system, I'll probably pull out some models, but I don't know if I will with this. We may just stick with the PowerPoint slide. Okay. So let's talk about the ear. Okay, one of the special senses, the ear. First off, we have a lot of anatomy here, and we know that the nerve for the ear is the vestibular cochlear nerve. And the vestibular cochlear nerve is actually split into two nerves, the auditory nerve, which is also called the cochlear nerve, and the vestibular nerve, which is more with balance and helps with vestibular balance, okay? And they're a branch off of this cranial nerve, the vestibular cochlear nerve, all right? So really important that you know that, okay? And that branches off because our ears do two things. Number one, we can hear. Thankfully, you can all hear me as we talk, okay? And then number two, this actually helps us balance. In these semicircular ducts, okay, or the semicircular canals, there's tiny little crystals, all right? And these crystals, as they shift and move, send a signal to the brain and help us correct our balance. Sometimes these crystals can become dislodged, and when they become dislodged, we get something called benign perioximal positional nystagmatism, or benign perioximal positional vertigo, okay? And you actually, part of your assignment for this lab, the ear lab, is to read an article on BPPV, or BPPN, that's the acronym for it. And you got to just write a brief explanation on what that is, okay? But that occurs here in our semicircular canals or semicircular ducts, all right? So that's the innervation, and that's how it plays a role. Really, really important here. Just like smell and sight, hearing is a really important sense, okay? Some other important things that we can talk about here are tympanic membrane, and then the ossicles, and we'll go over these ossicles here in a minute. These are actually bones, little tiny bones that help as sound vibrations come in, hits the tympanic membrane, sort of like a little drum, right? Hits that, and then these help to pass along the vibrations, which then go into the cochlear, and then voila, we interpret that, and our brain is so fantastical, puts it all together as, hey, that's sound and tone, okay? And different tones and different sounds are picked up from different areas. All right, the outer ear, okay? the outer ear here, also known as our auricle or pinna, okay? This is the anatomy of the outer ear, all right? And it's designed in such a way with these curvatures in very, you notice almost everybody's ear is roughly the same. It does this so that as sound comes in, sound reverberates and is funneled in. So kind of like the eye has a lens, well, the ear has a shape to it that helps to funnel sound in. Really freaking cool that we were designed like that. I think it's pretty neat. All right. One flaw with this, though, is in the ear canal, this area here, we have a little divot. And in some people, this divot goes way down. And if you use Q-tips, you're just pushing earwax in and then down, and it eventually can fill up this area. And that can actually block our hearing. So then you got to go to an ear, nose, and throat doctor and get that specially cleaned out. Okay. All right, same as your eye lab. Open up your ear lab, and you got to go ahead and start answering or matching some of these. So there'll be like an A here, so on and so forth, a B. You know, I don't remember exactly what it is, but you'll have to do that as well. Okay, I believe in your lab sheet, this is called the auditory nerve on your lab sheet, but know that it's also called the cochlear nerve as well. Okay, and together, remember, they're part of the vestibular cochlear. But go through, match all this stuff up here. Okay, you can see the eardrum, also known as the tympanic membrane. On your lab sheet, this will be called the tympanic membrane. So this right here is the tympanic membrane. Eardrum is just sort of the layman's term. Here we have from our ear canal, once we get into this chamber here, there's a tube that comes down. This tube comes down our face, kind of back into our nasal cavities. So if you have a runny nose and you're blowing your nose a lot, sometimes you can blow mucus up into your eustachian tube. And if you blow too much mucus up into your eustachian tube, you can actually cause yourself to have an ear infection, right? When you're in a plane and you're trying to clear or pop your ears, it's this area right here that you're trying to equalize pressure through your eustachian tube. So if you have a cold and you go scuba diving, or if you have a cold and you go in an airplane, it's going to be a lot harder to clear or equalize the pressure because your eustachian tube tends to be blocked, okay, by mucus. All right, so right through here you'll have mucus. Okay, so remember the outer ear. Okay, our outer ear, we have our auricle. Okay, then we have our external auditory meatus. That's this region right here, our external auditory meatus. It's all this tissue here. All right, it kind of starts right from the temporal bone here, and then right here's our external auditory meatus. It's right just outside of our ear canal, the tympanic membrane. All right, now we're getting into the middle ear. All right, the middle ear. These three bones, we're going to zoom in here in a minute, but we have the malleus, okay, the incus is right here, and then the stapes. All right, so the malleus, the incus, and the stapes. All right, those are the bones that help transfer vibrations to the semicircular canals and the cochlear. All right. Our inner ear, the inner ear, right through here. So this whole region, so if I were to take this whole region right here, this whole region right here is called the bony labyrinth, okay? Because this is kind of bony tissue, but it's a labyrinth. The cochlea has basically cilia that comes through, and that's what collects the vibrations, all right? Let's see, maybe the next slide goes a little bit. Oh, this is just a view of the tympanic membrane. You don't need to know the anatomy of the tympanic membrane itself, but it's pretty fragile. You don't want to break your tympanic membrane or poke it with a Q-tip or something like that, okay? Because that would not be good. Oh, I think I forgot to put up that last image that I had showing the stapes, the incus, and the malleus better. But you can kind of Google that as well, or Google anatomical parts of the ear so that you can fill out your assignment. All right, so this is pathway. When sound is collected, so we're just thinking about sound now, not balance. When sound is collected, it goes through the cochlea, okay? Higher frequencies occur first, and as we go through, we start to go into lower frequencies and lower tones, all right? So if you think of the keyboard, you play the higher keys, that comes in first, and then the lower tones come in last. Interestingly enough, that's why higher frequencies sound louder to us, is because they get picked up earlier and interpreted earlier on rather than having to travel on through. Lower sounds sound just that. They sound lower in sound, lower in frequency. Okay, so the higher frequency sounds are early on picked up. Okay. Okay, and then remember, it's picked up by the cochlear branch. So when we have sound coming in, it's picked up from the cochlear branch, the auditory branch, or the cochlear branch of the nerve, joined together to the vestibular cochlear nerve, all right, and it goes right into the medulla oblongata at the cochlear nucleus. From the cochlear nucleus, it travels on up through a tract and goes to the inferior colliculi. Remember, that's at the posterior aspect of the head in the corpora quadrigeminum. All right. At this point, we can have a motor output if we want to, if it's reflex, right? If you hear a car crash, you move, you duck, you do something, you hear a gunshot, you kind of move, you duck. You don't have a conscious awareness of that. All right. So that occurs here. And we want that to occur relatively soon and quick as that information comes through. Information that we want to be conscious about from here then passes on to the thalamus, and then from the thalamus it goes on to the temporal lobe in the auditory cortex. Okay, and then you can kind of see how it's interpreted. Here's where we get the conscious awareness. All right, so a reflex will occur prior to that. All right, pretty interesting, I think. You can look at the ascending pathway on this side. Okay, as we come in from the vestibular cochlear nerve, where these connections are. I'm not going to test you on this per se, but you will have to write out the actual process that I just went over there. What is the auditory process? The basic and brief auditory process, okay? So it's basically this slide and this one as well. So this slide as well, okay? So here you can see just another good interpretation of when sound comes in, it hits the tympanic membrane, goes to the malleus, the incus, travels down to the stapes, helps to transmit the vibrations. The vibrations come on through, go through to the cochlear, okay? Gets picked up, high frequency sounds first, then lower frequencies, and then the cochlear nerve picks it up, joins the vestibular cochlear nerve, goes on into the medulla oblongata, into the cochlear nucleus, and then moves on up to the, the superior olive, but then also to the inferior colliculus in the corpora quadrigeminum, and then the thalamus, and then on to the auditory cortex, all right, in the temporal lobe. That's basically the pathway right there. In a brief version, I should say. All right, so what about our semilunar ducts? This is an example of BPPV, benign perioxysmal positional vertigo, all right? Remember, like I said, there's crystals that are in the semilunar canals, all right? And these crystals can become dislodged. If they do, you need to do a maneuver to get them corrected. Usually it's older patients that have this, all right? My mom suffered from this. It's not fun. You get really bad vertigo. Your eyes dance. It's called dancing eyes, and you can get kind of dizzy and fall. This is really high-level stuff. This is what an ophthalmologist learns or a PA learns for an ophthalmologist. You don't need to know this slide here per se for your test. I just wanted to show it to you as an example. You're going to be reading about BPPV, and and learning all about it. This is 2 other treatment options right here. This is how you diagnose it. Okay, you put them in this position here, and whether their eyes dance to the left or dance to the right or rotate around, it's all because of a reflex that's occurring and vertigo that's occurring because of these crystals that are dislodged. All right. And then here's the treatment. Here's one of the treatment maneuvers for it to try and get those crystals back into place. I know it's really crazy to think. I never heard of this until my mom got it, and then I learned how to treat it. It's called the Epley's maneuver. There's several maneuvers to do it, but pretty interesting stuff. So this is what can go wrong with your balance if those crystals become dislodged in the semicircular canals. I think I said semilunar canals earlier. I was thinking about the heart. Semicircular canals.
The big old ear. Yep. Look at this guy. A little bit of a size difference between the eye model and the ear model. So here we have the outer ear. Remember, like I said, everyone's ears are designed in such a way, and you don't need to know all this, but you know, the superior aspect, which it's kind of hard to see, I have to lift it up. The superior aspect is the helix, starts on the superior helix and comes around. This is the helix. This is the lobe. All right. This is the anti-helix, the anti-tragus. This is what this is known as. This is the intertragic notch. Okay. This is the tragus over here. But you don't need to know all these little things, you know, anti-helix, and then the triangular fossa is up through here, so on and so forth. What you need to know is why this is designed the way that it is. Okay, it's designed the way that it is so that sound passes on through and funnels down into the canal. Okay, and then there's the canal. Okay, and we want sound to funnel through the canal. Now, some people, remember, like I said, some people, their temporal the temporal bone kind of dips down here a little bit, and that can tend to cause them to have earwax buildup underneath. I want to like unpop my ears when I talk about that. This tissue here, this cartilaginous tissue here, we have the external auditory meatus. Okay, it's all through here, the external auditory meatus. All right, and then our ear canal through here. And then if I'm sound, if my eraser's sound, I'm coming through and I'm gonna boom, and I'm gonna hit the tympanic membrane. And if I take out the tympanic membrane, we can see the tympanic membrane, and just on the posterior aspect of the tympanic membrane is one of our first ossicles. Okay, and remember that those, I'm going to set that back in, but the ossicles, there's 3 bones. Okay, and remember that those 3 bones are the basically the first one that makes the contact is the malleus, the incus, and then the stapes. And the stapes comes on down and helps to transfer basically all 3 bones help to transfer sound or vibration into this inner portion, this middle portion of the bony labyrinth. Okay. And I'm going to go ahead and pull that out right now so we can name those basically name those anatomical parts. Okay, I'm going to go ahead and take the bony labyrinth out, and then I really want to just go into detail and name this stuff. So if you have your lab sheet, we can really dive deep into the bony labyrinth itself, which is also known as the inner ear. Okay, so you can see how the semicircular canals, this is what holds the crystals. Okay, and that vestibular nerve comes and attaches onto the semicircular canal. Okay, and the crystals inside here are what help maintain balance of the body. It's just one of the many ways that we maintain balance. We maintain balance from these, the semicircular canals. Vision helps us maintain balance. And then we have tiny little cells throughout our body called proprioceptors and mechanoreceptors, and those also help maintain balance. But here the crystals inside the semicircular canals help to maintain balance. The stapes, that is a part of sound actually coming through, the stapes comes right into this middle portion right here, right before the semicircular canals, and then just before the cochlear. This middle portion is called the vestibule. Okay, So this is the vestibule. That's on your lab assignment sheet. The vestibule is the middle portion in between the semicircular canals and the cochlear. And this is the whole thing is called the bony labyrinth, okay? And then we have the the cochlear nerve or the auditory nerve right here. It's called both, right? And then this is the cochlear here. And then remember that vibrations come through and wrap around, and these vibrations are picked up by these tiny little cilia-like hairs in this structure. And these cilia, or these nervous tissue are just basically lined up like so. And they pick up the vibrations, high frequencies here, lower frequencies as you get deeper inside. All right, but that is the bony labyrinth or the inner ear right there. Okay, and it kind of just sits in just like that. So we can kind of get, I'm going to hold my tympanic membrane because it's going to fall out if I tip it, but kind of want you to see a little top-down view there of how it all sits in. So all of this sits in the temporal bone of the skull. Okay. What's this long tube right here? If you remember, this is the eustachian tube, okay? That's the eustachian tube. This goes to our nasal cavities, part of our nasal cavities in our face, and this is what we help to clear pressure in our head when we go scuba diving or in a plane or in the mountains or coming out of the mountains, right? Any sort of pressure differential. If you're sick and you blow your nose too hard, remember, mucus can come up into here and can cause an ear infection in here. Bad news bears, not good. All right? Okay, so basic, real brief overview of this ear model itself. I know we can't go in too depth in it, but like I said earlier, the brunt of it is going to be in your PowerPoint, so make sure that you have your lab sheet open and knocking both of these out as you go through it.